Good day, everyone. My name is Brian Jones, and today I will be giving you a presentation over stem cell differentiation and its potential uses in the treatment of diabetes. Now, before I can get into the real meat of my presentation, I will have to cover some minor background information. While I know that most of you are probably already aware of most of what I am about to say, there might be a few of you who are not, so please bear with me during this time. Diabetes is one of the more prevalent diseases that America has in this day and age. There are two different types, type 1 and type 2 diabetes. They both work differently, but they have the same base cause, which is the body's either inability to produce or use insulin. Type 1 diabetes is the version in which the body can no longer produce its own insulin, and we already have decent treatment methods for that, which is just to inject insulin manually and let your body process the insulin that is injected as the insulin that you use. Type 2 diabetes, on the other hand, is much more problematic. Type 2 diabetes is the version in which your body can't use insulin anymore. It produces it just fine, but it cannot use it anymore. We don't quite understand how or why it does this. We do still have some treatment methods for it. These usually involve taking drugs that allow you either to get rid of or store the extra glucose that you produce while eating. Though there are a few that do aid in the production or the use of insulin by the cells that are resisting their current use. Now, these help, but they're not a surefire cure for type 2 diabetes. They help manage it more than anything, unlike type 1 where you can inject insulin. It's pretty much it, you're set, because the only thing that type 1 needs is the insulin itself, while type 2 has to go around the fact that it can't use the insulin anymore. There will be a little bit more over this later, but that's the basic information on diabetes. The cytoskeleton of a cell is probably something we are all intimately aware with at this point, with all of the classes throughout school and college that cover it. But there are some that believe that the actin has something to do with cell differentiation. So we will be covering it a little bit right now. So the cytoskeleton is the structure that gives the cell its shape and its ability to move as well. The actin itself is a specific filament of the cytoskeleton that motor proteins use to transport molecules and cellular bodies through and across the cell cytoplasm. With all of that in mind, let's move on to cell differentiation itself. Cell differentiation starts at the embryo. As the embryo first starts dividing, it's still dividing into totipotent stem cells, which have the ability to differentiate into all three of the different stem cells for germ layers: ectoderm, mesoderm, and endoderm. From there, the different divisions keep differentiating more and more and more until you get the specific cells and tissues. They do this because of a few different hormones and molecules that the cell produces. These all combined make up a certain gradient that makes it to where each and every different organ and tissue forms exactly where it is because of the specific hormones and molecules and even cells around the cells that are being. As such, with the ability to create our own stem cells, we would be able to potentially fix, if not replace, any defective or destroyed tissues or organs that we have in a specific person, as long as we are able to derive their stem cells from their own cells so that they don't reject the organs and tissues that we are putting in and on them. With that knowledge in mind, the use of stem cells in the treatment of diabetes, while being effective for both types, is actually more useful for the second type, because if we could find a way to, as I said before, replace the cells, we could make it to where it goes from being a type 2 diabetic, which you can manage the symptoms of but not really treat. In that case, we would finally have something like type 1 has in insulin. That being said, it could also be used for many other diseases, including type 1 diabetes, because if we could just replace the organs and the cells, then we wouldn't have to worry about those anymore, as long as those cells that we replace them with can divide and proliferate themselves. Now, with all of that being said, we can finally move on to the main portion of this presentation, where we talk about the two papers in question that I am covering. The first one is a paper written by Cheryl Southard and their compatriots, titled Generation and Selection of Pluripotent Stem Cells for Robust Differentiation to Insulin-Secreting Cells Capable of Reversing Diabetes in Rodents. Now, this, of course, is more geared towards type 1 diabetes, as while it can help manage type 2 diabetes, it will have more limited success for that. In this paper, they use the more standard practice of taking adult cells and then using IPS reprogramming factors to turn those fully differentiated adult cells back into stem cells, or IPS cells. And from there, they can redifferentiate them into the different cell types based on what they program the cells with molecularly and hormonally. Now, they don't use cells from the individual in question, but rather from a lineage of cells that has usually been created beforehand and are just being redifferentiated again to become what they are needing in this case to help treat the affected organ, in this case the pancreas, for diabetes. Now, in order to differentiate these cells into their pancreatic insulin-secreting counterpart, they needed to treat them with specific hormones in order to set the gradient needed to reach that point. In this case, those were HSA, 127 supplement, Activin A, Wortmannin, DMIM-F, retinoic acid, KGF, noggin, cyclopamine, secretase inhibitor, glutamax, nicotinamide, IGF-1, and BMP4. From there, these now differentiated cells were bathed in different concentrations of glucose and potassium chloride to see what their response was to said masses of glucose. Then from there, the cell lines that had the best response to the glucose treatment then proceeded to be injected into rats that had their ability to create insulin destroyed, whereupon the lower levels of blood glucose were detectable within 7 days of the injection and lasted a period of a few weeks. These cells in particular were found to have 60% of the effectiveness of regular insulin-producing cells, which, while not being perfect, is definitely a good direction. From these results, though, it is still clear to see that our methods of producing these insulin-producing cells, or in this case, differentiating cells into insulin-producing cells, can still be improved and built upon. In the end, though, this line of cells was still marked as suitable for use in the treatment of diabetes and set aside as a stem cell producing. Now, for the next paper, it is a paper by Nathaniel Hogrebi and their compatriots titled Targeting the Cytoskeleton to Direct Pancreatic Differentiation of Human Pluripotent Stem Cells. The cells that they used were taken from 4 different stem cell lines and a BJFF6 line, and for their controls they used human events. They used 2 different protocols in the differentiation of these cells in order to help check the collagen-1 levels in different cells and what the resulting expression of those were. Because collagen-1 is produced by the actin filaments on the outside of the cell. From there, the actin ratios were determined Western blot. Through their experiments, it was shown that the levels of NeuroG3 in KX2.2 and NeuroD1 were lowered, whereas the SOX9 expression was raised. This was attributed to the lessening of the transport of the NeuroG3 in KX2.2 and NeuroD1, because SOX9 is repressed by those molecules. Now, with that knowledge, it could be possible to combine these 2 ideas into one experiment. Say we were to make a cell specifically for the production of insulin like they did in the first study, and then alter its expression of collagen as well so that it can bind less to those molecules that suppress SOX9 or more to those molecules that suppress SOX9 so that more insulin is able to be produced. And as such, we might be able to increase our productivity of insulin to a more natural level, depending on if that is one of the issues with our current methods of producing insulin-producing pancreatic cells from redifferentiation.